It's my delight to give you a very short overview of just some aspects of how we're using materials in the field of tissue engineering. So I thought what I'd do is start off by thinking a little bit about how materials have been used over time in the body. And start by showing you this picture of this beautiful nacre shell. Now this used to be used by the Mayans as an artificial tooth. And it's more recently when we've been able to actually study the microstructure of this material within this field, that we can see that it actually has this beautiful microstructure composed of these platelets of inorganic material that are lodged also with some organic material. And so it turns out actually that this microstructure is very good at interfacing with bone, and so these artificial teeth, right back in the Mayan times, could integrate into normal jawbone and be used functionally. Now, over time, there's been many other examples of the use of materials in the body, and this is one of my favorite ones. This comes from World War II, where Sir Harold Ridley, who was an ophthalmologist, used to see pilots coming back from Spitfire missions. And what he noticed is that the pilots had shards of material in their eye that was coming from this plastic canopy of the airplane. And the interesting thing is that those shards of material were lodged within the pilot's eye, but they weren't causing any inflammation at all. So Sir Harold Ridley was able to isolate this material. He was able to show that it was this plastic material, and was able to then push the development of this as the use of the first intraocular lenses, which are now used by millions of people worldwide. Now, in those first examples that I've shown you, and particularly this one with the Spitfire canopies, the material was actually used in the body because it wasn't causing any inflammation. And if we look now at the field of tissue engineering, where we're using materials within the body, you can see here a classic schematic of that process. The idea is that we take a material system and we might grow some cells on it, for example, stem cells from the patient. But rather than have the material be inert, we're very actively looking for it to be bioactive. So we want the material to interact with the body, and we also want it to dissolve over time so that we're left with healthy repair tissue. So this field of tissue engineering is really touching upon every tissue, more or less, within the body. You can find applications in this area. And this is just a few selected examples from the bone regeneration field, and you'll find so many different types of material systems that are used for repair within the body and, for example, within bone. And these can span from polymer-based systems to inorganic systems, from nanofibers to microstructures, all sorts of different materials characteristics. And some of the things that people are thinking of is how can we incorporate chemistry within this field, and how can we make materials with very tailored bioactivities. So this just shows you a schematic from a review paper that we developed a few years ago that shows you some of the important things. So you might think, for example, about adding bioactivity to your material, adding it artificially, and that might come in the form of small peptide molecules or delivery of nucleic acids, all sorts of interesting chemistries and biochemistries going on there. You also might think more fundamentally about how do you change the hydrophobicity of your material, how do you activate different signaling pathways within cells by designing your materials in a tailored way, and how do you control what their mechanical properties are like and how quickly they're going to degrade. And again, you might be controlling at the nanoscale, but how do you then make this into a 3D material with different properties, whether it be fibrous or porous? And these are all different aspects that are being explored very vigorously within this field. But one of the key things that we think about a lot is how much we want to biofunctionalize our materials, because as you can see in this schematic, you can start off with very simple material systems at the top here, and these can work in certain situations, but often they don't necessarily provide enough information to the cells and to the developing tissue. So you might want to put in more and more complexity to your material. But one thing to realize is that when you put in this extra complexity, you can increase potentially your bifunctional specificity and make the cells more likely to form the right kind of tissue, but you can also potentially make your materials very difficult to translate into the clinic. And in this field, a lot of the aim has to be about making materials that eventually will make it into patients. So what I wanted to also highlight to you is actually some very simple materials innovations that you can make that can be very effective in regenerating tissue. One of those simple things we might think about in terms of tailoring the chemistry of our materials is something very simple, like introducing some bioactive ions into the material. And this is a very active field. We, together with colleagues at Imperial College London, have been introducing ions such as strontium into materials. And you can do this rather simply, but you end up with a material that actually works very well for bone repair because of the bioactivity of strontium. And so even though strontium is a very simple ion, we know within the field it's good for bone repair. And what we've been looking at more closely is some of the gene networks that are activated by this strontium to try and understand better how this material is so good at bone repair. And this is just from one of our recent papers, where you can see in this Venn diagram that many different genes are activated within human stem cells once you add strontium to the material, and that many different gene pathways are upregulated. This just shows some of the more important ones here, and you can see at the bottom some of these sterile biosynthesis pathways that essentially are going to be important in cell signaling and that help us to understand why better bone formation can occur with these types of materials. Similarly, you can think about changing the nanostructure of your materials to get tailored cell responses. And this is some lovely work from Matt Dolby and Richard Arefo's group from the UK, where they've created surfaces that have nanoscale pits on the surface. And in this very elegant experiment, what they show is that if you have those nanoscale pits in an ordered pattern on a surface versus a disordered pattern, so the same pits but just slightly disordered, and you can see that nicely in this picture here. If you then culture human stem cells on them, then that slight disorder will actually make those stem cells much more likely to form bone-like proteins than on the ordered pattern. And so that's just a very small change at the nanoscale. The chemistry of the surface is not changing at all, but yet you can influence your cells to form a more bone-like tissue, which is very exciting indeed. And many other types of nanostructuring have also been looked at, and I'm not able, of course, to do this huge field justice in this short talk, but one of the things that's quite interesting is different nanostructuring of surfaces, for example, in the form of nanopillars on surfaces, and this is quite an active area. You can see here a figure that's taken from a lovely review article from Karen Martinez's group, and this just shows some of the different nanopillar type architectures that have been made using either top-down or bottom-up methodologies, and all of these ones have been made particularly with the idea of thinking about how we can use them to interface with cells. Now, we've also been working a little bit in this field, and I'm going to just show you one example of how we've actually been making nanostructured needles to help in the tissue regeneration process. So this is really using nanostructured and microstructured materials to help in tissue regeneration. And these nanoneedles were developed by Ciro Ciappini, who's a research fellow within our group, and this was done in collaboration with Professor Taschiotti's group at HMRI, and you can see here a cell interfacing with these nanoneedle structures that we've made. Now, if you look at these nanostructures over time and how cells interact with them, you can see at this early 1 hour time point here, cells are slightly balled up on the surface, but you can see this nice interaction here between the nanoneedle and the cell. And then over time, cells will actually spread on the surface, and you can see this very nice interaction here between the needle and the cell. And this is just the nucleus moving out the way. If I show you a focused ion beam sectioning of this cell and then a false color reconstruction, as hopefully you'll see in this video here, you can see the needles in blue, the cell membrane is in purple, and then the nucleus is shown there in yellow. So the needles are actually not penetrating the nucleus, but they're interfacing nicely with the cell. And you can just see here a false color reconstruction of one of those focused ion beam images, and you can see this porous structure of the nanoneedle. Now that porous nanoscale structure is really exciting because it means that we can think about loading things into the needle and then delivering them to cells and tissues. Now, that would be important for many different reasons, and the example I'm going to show you of why it's relevant in tissue engineering is that in the field of tissue engineering, one of the issues you often have is that it's if you implant an engineered construct into the body, very often the host blood vessels can't integrate well enough or quickly enough with the implanted engineered material. Now, if we were able to regenerate blood vessels at the site of implant before we implanted our tissue, we believe that could be extremely important in tissue engineering. And so we thought about using these needles to deliver to muscle some nucleic acids that encourage the formation of new blood vessels. And this just shows you a control image of some muscle where we've just dropped some nucleic acids onto the surface without using our nanoneedles, and not too much happens. Now, if we take our nanoneedles and we deliver them to the muscle, we deliver some nucleic acids that can encode for vascular endothelial growth factor, which is a growth factor that's going to help blood vessels forming. We can see that even at 7 days, we start to get lots of new blood vessels forming on this muscle in vivo. And then over time, those blood vessels get more and more mature. You can see the quantification here, and you can start to get a really good prevascularization of the site. So this is really important, I think, in this context in terms of making new blood vessels, but it also opens up the possibility of both this work and lots of other work within the field of using nanoscale materials and nanoscale delivery systems to actually deliver proteins and nucleic acids to tissues so that we can help in the regeneration process. So this is what I wanted to end on. I'd like to thank you for your attention, and particularly thank the collaborators of this work. And obviously I've only been able to show a couple of examples from the field, but hopefully has given you a bit of an overview of some of the use of material science and materials technology within this very broad field. Thank you.